Our project is immune response pathways and gene expression dynamics in PBMCs of malaria-infected individuals versus healthy individuals. For our abstract, this project studied genetic and immune system differences between people with malaria and healthy controls. Using GO2R, we analyzed blood samples and identified key genes with increased activity in cerebral malaria, such as MMP8, ARG1, and BPI. Visualization tools like volcano plots and Venn diagrams helped us understand these changes. This research highlights molecular mechanisms in malaria and could guide the development of better treatments in the future. For our introduction and background, malaria is a life-threatening disease caused by Plasmodium parasites, spread through bites from infected female mosquitoes. Once in the body, the parasites travel to the liver, then enters red blood cells spreading through the blood. Malaria is treated with medications based on specific Plasmodium species causing the infection. Understanding these processes helps develop better treatments and prevention strategies.
For our hypothesis, we stated differentially expressed genes in peripheral blood mononuclear cells of malaria-infected individuals compared to healthy controls are enriched in immune response pathways, including cytokine signaling and T cell activation, potentially highlighting genes involved in immune evasion and pathogenesis of the malaria infection. We chose this as our hypothesis because we figured that if something irregular was happening in the body, obviously genes would be overexpressed or underexpressed due to the situation.
For our methodology, we analyzed gene expression in malaria-infected individuals compared to healthy individuals. We pulled this data from a publicly available data set called Whole Blood Transcriptome of Childhood Malaria, GSE 1124. Using the GO2R analysis tool, we identified differences between healthy and infected individuals by creating volcano plots and Venn diagrams. The analysis grouped individuals into 5 categories: healthy, asymptomatic, uncomplicated, severe anemia, and cerebral malaria. We then cross-reference identified differentially expressed genes with known gene functions to further understand the processes involved in malaria. As we can see, looking at the graphs, our volcano plot was showing upregulated genes in red and downregulated genes in blue in cerebral malaria patients compared to healthy individuals with all the significant genes highlighted. Our Venn diagram illustrates overlapping and distinct differentially expressed genes across various compositions, such as cerebral malaria and versus healthy, or severe anemia versus cerebral malaria. As we can see, in cerebral malaria versus healthy, there were 873 differently expressed genes. In figure 3, we can see that the ARG1 gene was very upregulated in individuals with cerebral malaria. In figure 4, we can see a similar thing. The BPI gene was highly upregulated in individuals with cerebral malaria and severe anemia. And the bottom 2 figures are just the chromosomal locations of those 2 genes discussed.
For the results, we used the GO2R tool to analyze gene expression in 5 groups: healthy, asymptomatic, uncomplicated, severe anemia, and cerebral malaria. The analysis revealed key differences, with the biggest changes seen in the upregulation of MMP8, ARG1, and BPI genes in individuals with cerebral malaria. These findings confirm that cerebral malaria has a distinct gene expression profile compared to other groups, highlighting potential targets for future research and treatment strategies.
For our conclusion, we found that certain genes like MMP8, ARG1, and BPI are more active in individuals with cerebral malaria. These genes are linked to inflammation and immune dysfunction, highlighting their role in severe infections. The results suggest that cerebral malaria may have a distinct molecular signature, which offers insights into the disease's progression and potential new targets for treatments and vaccines. Future research could focus on how these genes influence malaria and help in developing better therapies.
There we go.